Hello dear friends. Today we are going to study or going to discuss the next point of the unit number 4 is Pearl Oyster culture. Before the Pearl Oyster culture, in a previous lecture we have discussed the freshwater prawn culture, and today we are going to see what is Pearl Oyster culture. So throughout the centuries, pearls have been associated with wealth, femininity, purity, wisdom, patience, and peace. And due to their appearance, they have often been compared to the moon and are said to have a calming, protective, and strengthening effect on the reader. So nowadays, these pearls are still regarded as a beautiful and feminine gemstone. So today will discuss the pearl oyster culture, how we culture the pearl. Okay, so pearls are formed as a natural concretions secreted by the mantle within the shell of the bivalve mollusk called the pearl oysters, and it is the same material as that of a shell of a mollusk. So the mantle, as a protection against a foreign body, usually secretes them, and a pearl is formed of the nacreous substances deposited one above the other in a concentric layer laid down by the epithelium of the mantle surrounding the nucleus, and it shows a central nucleus around which several layers of organic and inorganic materials are laid, and the pearls are usually spherical in shape. Okay, usually in a market we will see, or we have seen the pearls, their shape is spherical in shape. So the chemically, when we discuss the pearl, so when we analyze these pearls, the chemically analysis of a pearl shows the presence of CaCO3 90%, calcium carbonate, organic substance 5%, and water is remaining part, that is the 5%. Okay, and the pearl oyster, it belongs to the genus Pinctada of the family Pteriidae under the class Bivalvia. Okay, so they are marine in habitat and are present in coastal waters globally. Worldwide it is present, and certain species of freshwater bivalves occurring in natural freshwater bodies like lakes, reservoirs, rivers, from small sized pearls as marine pearl oysters. Okay, and the pearl is secreted by whom? The pearl is secreted by the mantle of the pearl oyster as a protection against foreign object, which may be sand particle or minute larval forms or other such things. Okay, and after that the pearl, the oyster is secreted, the secretion over the particular foreign body, and after some time, after some year, the pearl is formed. So see here, to study the mode of formation of pearl, it is essential to know the structure of shell and mantle. So shell of the pearl oyster is composed of three distinct layers, three different layers. We have this here, this is the shell exterior, shell interior, and the nacreous layer. So these three distinct layers is very important. So see, first is the periosteum. Periosteum, it is the outermost greenish. Okay, it is the outermost greenish brown thin translucent layer made up of an organic substance called conchyline, and it is secreted by the mantle, and it serves to protect the underlying layers from the harmful effect of weak carbonic acid in the water. Okay, now the next layer is the perismatic layer. So it is the middle layer secreted by mantle, and this layer is made up of minute crystal of calcium carbonate, and it gives strength and rigidity to the shell. This perismatic layer, it gives strength and rigidity to the shell. Okay, and last layer, that is the nucleus layer. It is the innermost, this is the innermost layer of the shell, and is better known as mother of pearl. See, this is the place we can call the mother of pearl, and because this layer is responsible for the formation of pearl, that's why this layer is called mother of pearl. Okay, now it consists of alternate layers of calcium carbonate and conchylium, and this layer is also secreted by mantle, and its function is to protect a delicate surface from harmful effects of the foreign particles. Okay, and that pearls that are formed due to the irritations caused to the mantle by entry of foreign bodies. So pearl is nothing but a foreign particle is entering into the shell of the oyster. After entering this foreign particle into the shell of oyster, the oyster is secreted some secretion over the foreign particle, and after some time layer and layer, so the foam is formed. Okay, so whenever any sort of foreign body enters and gets between the mantle and shell, it becomes enclosed in a sac of mantle epithelium. Okay, so this is the place where we can say this mantle. So whenever any sort of foreign body enters and gets between the mantle and shell, it becomes enclosed in a sac of mantle epithelium, and this foreign body now act as an irritant. So whenever this foreign body enter into the pearl oyster, so when it enter into the mantle into the pearl oyster, so it act as an irritant and stimulates the mantle epithelium to secrete concentric layers. So this is the mantle cavity. So this mantle epithelium is secrete concentric layers of nacre around this foreign body. So these foreign bodies enter into this cavity, and after entering into the cavity, so it act as an irritant and it stimulate the mantle of this mantle epithelium to secrete the concentric layer of nacre around the foreign body. And these layers when harden, okay, means these layers when hardens, number of layers is put by this cavity on the particular foreign body, and when it hardens, now it becomes pearl. Okay, and size of this pearl is directly proportional to the degree of irritations caused by foreign agents. How much irritants is caused by the irritants, and that is depend on this we can say the size of the pearl. Okay, so time taken for the formation of pearl of average size is 3 to 5 years, means here. So a single perm is formed in between 3 to 5 years. It requires 3 to 5 years to form the pearl in this oyster. Okay, now this is the biology of pearl. So see, we will discuss now the biology of pearl oyster. So the oyster usually breeds several times in succession. The larvae are free swimming and feed upon microscopic organisms. So it is filter feeders and it feeds upon the microscopic organisms. They undergo metamorphosis, number of metamorphoses, and finally settle at the bottom in large numbers. Okay, and the young oyster has a fast growth rate and it attains about 50 mm size in a year. Means in a single year the young oyster has reached up to 50 millimeter in size. So this pearl oyster feed on small algae found in the water column through filtering large amount of water. That's why they are filter feeders. Okay, they are large amount of water, filter the water, and after filtering the water they get some small algae for their as a food. Okay, now the pearl culture process. The process of pearl culture includes the following steps. The step 1, construction of pearl farm. It includes 2 steps. Number 1, selection of farm site. A site is selected that provides constant temperature, protection from waves, shallow waters. B, construction of pearl farm. Wooden rafts of 5 into 5 meters are placed in a sea water using proper anchors. Lines of ropes are hung on these rafts. So see, the selection is more important. The selected site provides constant temperature, it protects the waves, shallow waters. So when we select the site for this pearl culture, these things are very important. Okay, and number two, the construction of pearl farm, how it concerned. So wooden raft can be used for this. We can say this is in this picture. The construction 5 into 5 meters are placed in seawater using the proper anchors. We have used the anchors for fixing the wooden raft in the seawater, and lines of ropes are hang on these rafts. So we can see in this picture. So this is the lines of these ropes. Okay, and they hang to the raft. This is the raft. Okay, this is the drum which for this it is useful for the flowing fluid. Okay, so this is the bamboo raft, and these ropes with the oyster attached, they are hang into the water body. Okay, into the water body. Now next step is collecting the oysters. After the construction of pearl farm, when we number one, we select the site for the pearl culture. Number two is the construction of the pearl farm. Now the collection of oysters. So after the construction of pearl farm, the divers set off the bottom of the sea to collect the oysters. So oysters are generally located on flat rock bottom, and the shells collected are clean. First they are clean, then again they are sized, means a small, medium size, big size, they are categorized and placed into the basket for the storage until they are transferred to the pearl farm. You just collect this oyster from the seawater, and after collecting, we have to clean it, then we have to size it, and we have to store in a basket until they are transferred to the pearl farm. And the larvae of this oyster can also be collected, and that process is called a spat collection, and these larvae are again grown up to 2 years so that they can be seeded, introduction to the foreign particle. Okay. Now number 3, the seedling. Step 3 is the seeding. 2-3 year old healthy oysters are considered for surgical implantation, known as seeding. Seeding means what? So after 2 to 3 years, the oyster can be collected, and that is used, or that can be considered for a surgical implantation, and that surgical implantation is known as seeding, and this is a very delicate operation and involves 3 stages. Okay, then the preparation of the graft. A donor oyster is sacrificed to obtain mantle. Then mantle is needed by the host oyster to accept the nucleus. So nucleus is a foreign particle, usually sand or a glass particle. Now the attaching the graft. The oyster is open with a special wedges and pillars. Then the sand or the glass particle is introduced between the shell and the mantle, and the oyster is then returned back to the water. So attaching the graft, so the oyster is opened with the special wedges and pillars. Then the sand or the glass particle is introduced between the shell and the mantle of the oyster. Then after this attaching the graft, the oyster is returned back to the water, and the inserted core irritates the oyster, provoking it to gradually coat the core with thin layer of mother of pearl nacre. And after some time, the oysters are collected, and they are see whether the implants have been accepted or not. Okay, an oyster which have rejected the implant are returned to the water and are once again operated. Okay, and the oysters which have accepted the implant, they are transferred to the pearl farm for the further procedure. Okay, now the step 4 is the caring of oyster. Okay, so it is very important how much care we have taken to the oyster. So the shell which have been grafted are transferred to the pearl farm, placed in a basket or ropes attached to the floating rafts. Just we have seen, the oyster can produce more than one pearl in its lifetime. So number 5 step is the harvesting. After these 4 steps, the number 5th is the harvesting, and after 2 to 3 years, it requires, we have already discussed that the 2 to 3 or 2 to 5 years, the pearl is formed. So after 2 to 3 years, the oysters are harvested, and it is necessary to make a trial harvest to determine whether the pearls have sufficient coating. After if we not taking the trial, or if it is not sufficient, then an additional 6 months to a year for culturing is necessary. If we are harvest the pearl within the 2 or 3 years and we are not satisfied that there is no sufficient coating, so we are again adding to number of months, means 6 months or a year, we can put the oyster into the water for good quality of pearl produced. Okay, wait until the means the number of sufficient coating is there. Okay, so the oysters are spilled open and pearl bags are cut by the scalpel to remove the pearls that are formed into the pearl oyster. Okay, now the sorting of pearls, the last step. There are many different steps involved with this sorting of pearls. Firstly, the pearls are sorted according to their size, how much size of the pearl is formed, so small, medium and the large. Then the color, depend on the color of this pearl, okay, and the luster. They are the marketed later on. Okay, so this is nothing but the pearl oyster culture. Okay, so with this I'm stop here. Once again, if you have any query regarding this lecture or any previous lecture, so you just drop your question into our YouTube channel, so I can give answers, or I can I can give your questions answer. Okay, till then, goodbye. Thank you very much.